see you. All the best.
Thank you, sir.
Remove spotlight. Okay. So we will start in next one. Wow, there are a lot of schools who have joined. I'm really happy and excited to talk to you all. So before we start the session, okay, so it's 3:30. I would like to say hi to everyone. There are a lot of schools, and I'm really happy to
Hi,
see you
sir.
all. So what I'll do, I will mute everyone. I don't know how. Yes, so that all current yes. So I will unmute. If someone wants to talk to me, I will definitely unmute you all. Okay. So I hope you all are happy, healthy, and you are enjoying our sessions that we are delivering from IIT Gandhinagar. So before I start the session, I would like to ask you one thing. How many of you have the material that we asked for? I will launch the poll, so you can just answer yes and no. Okay. Uh, question. Okay. Do you have all the material for the sessions? Amazing. I will give like some time to all of you. Everyone, if it's possible, you can just say yes or no. If you have some material or you were not aware of the material you required, you can just. Good. You can continue the poll. I will randomly pinpoint someone. Wow. Hello, Shri Devi Kudumula. Hello, kids. Hi, ma'am. I can, you can unmute if you want. I just want to see there are a lot of students sitting. So, oh. Okay. So a lot of you have the material and few of you have some materials. That's fine. Okay. Good. Thank you, everyone. Nice. So as you can see, my name is Vikram. I am talking to you, all of you from CCL IIT Gandhinagar. It's in Gujarat. So in this session, I will share my whatever I have learned so far in biology. So during my session, I will take few sessions with you. So this session is about photosynthesis. So it will be around biology. So I hope you people like a biology subject. If you want, I will try to make you understand some basics about biology. So our today's session is on photosynthesis. So before starting this session, I would like to show you some of the slides, some of the PPTs, some of the like videos of the previous session that Aditi ma'am has took. So these are the schools who have made the thing that Aditi has mentioned. Everyone can see. If you can see, just you can thumbs up. You can see the slide PPT. Arey wow, amazing. So this school is from Andhra Pradesh, KGBB, Ponduru. So they have made the box that Aditi have mentioned, and this is the. Oh, just hold on.
मेरा नाम लुबना है। मेरे स्कूल का नाम केजीपी लोनी जिला गाजियाबाद है। आज हमने प्रोग्राम प्रोसेटिव प्रोग्राम में पोजिशनल बेस सिस्टम बनाया। मेरा नाम मंतशा है। मैं कक्षा 8 में पढ़ती हूँ। मेरे स्कूल का नाम KGBB लोनी, जिला गाजियाबाद। आज हमने पोजिशनल ड्रग सिस्टम मशीन बनाई है, हम सीखेंगे। पहले हमने एक रखा, एक का एक, फिर दो रखा, दो का दो, तीन।
So Aditi liked it very much. So she has seen lot of videos that you guys have shared, and she liked all the videos. She asked me to thank you all of you. So this is one more video from KGBB. Okay, why it's not playing? But oh, so like yeah. So many of you have shared the video with us. Thank you very much for sharing. So before starting the session, I would like to give a very brief introduction about what I do, what I really like. So as I mentioned, my name is Vikram. I am from Himachal Pradesh. So Himachal, have you heard about Himachal? Anyone, any school who heard about Himachal? Yes. So it is like it is very similar to Northeast. It's also green. Lot of mountains are there. I'm from Himachal, and when I was of like 7 or 8 years old, I used to do very crazy things. So is there anyone who collect different things? Is there anyone from you anyone who collects some coin, some insects or seeds? Anyone who likes to collect things? So when I was very small, I was 8 or 9 year old, I used to collect. I will show you what I used to collect. I used to collect these insects. The insects that you are watching here in your screen. I used to collect all these insects, and I used to bring all the insects to my home. I don't know who taught me to collect all these things, but I really like insects and collecting other things. So this is my hobby. This is my interest. Like from the very beginning of my life, I used to collect insects. I will show you few more other things. If any one of you also do these kind of things, you can mention your name in your chat box in the chat box. I am very, I'm very excited if someone of like any one of you is collecting the insects and other things that will, you know, reminds me of my old school days. So I used to collect these bees, wasp nests also. As you can see here, I have a lot of bees, wasp nests. I have these birds' nests. So this is my this is the thing that I really like doing, and I like to I like to make students understand biology through all these things. So this is my like interest. I really like. So is there anyone who used to collect all these things? Is there anyone from you? You can chat. You can add in the chat box if you want. So I will show you uh, a small slide. So okay. As you can see, so this is like I have a museum of my own. So in this museum, I have skulls, I have bees, wasp nests, insects, snail shells, seeds. So I have around 3 to 4 thousand specimens, and I carry all those specimens in my car, and I travel to different universities, colleges, schools, and I teach biodiversity to all of them. And I also teach biology to them. So this is my passion. I really like all these things. So any of you, so if you are like, if someone of you do all these things, you can write in the chat box. I can definitely interact with you after the session. I would really like to know if there is any kid or any girl who do all the things that I was doing, right? So moving ahead, so, so if any one of you, you can write in the chat box that I also collect all these things, or if you collect seeds, stones, anything you can mention. So we will talk later about that. Okay. Okay. So let's start the session. As you have mentioned, most of you have mentioned that you have all the things. So I will put a small slide, the things that you required for today's session. You need plastic glass, you need uh, syringes. I will also put all these things in the camera. Uh, I will put the things. So you need the straw. I will put, you need aluminum foil. You will need three. You can also keep, you can also take out your material uh, along with me. So you need all these three things. Along with this, you need a baking soda. It's very easily available in the market. You know, you need baking soda and you need leaves. It's better if you have spinach leaf, or if you don't have spinach leaf, you can use any other leaf also. Uh, Along with this, we need water. You know, tap water is okay. We need tap water and a light source. I can just switch on the light to show you that I have a light of yellow color. As you can see, this is a yellow light coming from this side. So I have a yellow bulb here. Along with this, we need few beakers. Okay. And if you have this liquid dish soap, it will help you. Okay. Great. So are you ready to uh, see what we will do today or we will learn today? Okay, great. So someone has mentioned PDPL KGBV Jula Pali. So they used to collect seeds, shells from the river or wood fossils. I'm really happy and I really wish you that you collect all these things throughout your life and enjoy the biodiversity or real biology. Okay. So in the today's sessions, what we will learn, so we will discuss, we will learn about the role of carbon dioxide in photosynthesis. What is the role or effect of light in the photosynthesis, carbon dioxide? We will understand the role of plants in our ecosystem. And as we all know, I think we all know that plant give us oxygen. So oxygen is a gas. So we will also try to see that in the process of photosynthesis, plants, they release oxygen, but we will not able to test whether through our experiment, today's experiment, whether that thing is oxygen or something else. So we will understand that plant release gas during photosynthesis. So we will learn about all these four things in our session. Okay. Wow. I'm happy that you are responding to me. So before starting this session, I would like to ask a small question to all of you about, yes. So the question is, how many of you have heard the term photosynthesis before? You can say yes or no. People who have joined us through YouTube, you can write it in the chat box, yes or no. Wow. So I think a lot of you have heard about photosynthesis. I'm very happy. Good. Yeah, it's a very, we talk about photosynthesis a lot. Good. Amazing. There are 130 schools. Let's wait for them. Wow. I think around 50% of you have heard about photosynthesis. Wow. Oh, there are few schools who have never heard the term photosynthesis. Okay. So for them, I will. Okay. So I'm ending the poll. Thank you very much. Wow. So to continue with this question, I am again launching a second question, which is what happens during the photosynthesis? The one you have answered that, okay, you know the photosynthesis, you know, you have heard the term photosynthesis, but what happens during the photosynthesis, I have uh, listed 4 options, whichever you think that photosynthesis is, you can wow. Most of you know that what happens during the photosynthesis. Plants make food in the process. Good. Nice. Okay. So some of you have not heard about oh, so great. So a lot of you know all of you know about, most of you know about what is photosynthesis. So I will just briefly explain. So plants, they make their own food. Have you ever seen any plant going to a market asking a shopkeeper, I am hungry, please give me some food? No, plants, they don't go to malls. They make their own food. So if you just sow a seed in the soil, later on it will automatically grow into a big tree. So the process through which that seed turns into a big tree is a process which is called photosynthesis, through which plant make their own food. So it's a very long process. A lot of things happens during the photosynthesis, but in the photosynthesis, plant make their own food, right? So we will learn a very small part of photosynthesis today. So okay, great. So before starting this session, uh, wow, I will launch one more very amazing question. I really like this question. So suppose if your school is planning to build a big garden, very big garden, and they have chosen 2 rooms where they will keep small saplings. So I will tell you what saplings are. The plants which are not that are still so I am showing you in the overhead what sapling means. Sapling is a very small plant that is not is fully grown. It's in the early stage. So sapling means that. So uh, okay. So what? So which? So your school is planning to build a big garden, and they have chosen 2 rooms. So these are the 2 rooms uh, where they will keep the small saplings until they will get matured, and they plant those mature plants in the field. So if you were asked to put those sapling in any of these 2 buildings, which one do you choose? A or B? Later on, I will ask you why you have chosen A or B. Wow, good. Lot of you are uh, giving amazing answers. There are only 2 answers, but yes, I'm really liking that someone is choosing A and B also. Great. Wow. So I am giving just like 30 more seconds. Then I will end this poll. Okay. So I'm ending the poll. Thank you very much. Okay. Okay. So, it's very, any of you who have choose the option B, can you raise your hand? Option B, which school has opted for option B? Okay. Option B, who has answered for, like, who answered for A? So I can see, Samsung SMT735. Okay. So you can unmute Mizoram kids with the yellow t-shirt. Like you answered A or B?
Langan ko, kunin todo ko.
A.
Ini ko.
And why? Why A? What do you think? Why you have chosen A? What was your reason? You can unmute. Wow. Any one of you can tell me why you have chosen A? Why you have chosen building A? What's the reason? Okay, so I will explain. Like, so A is the correct answer because plants, they need light to grow. In building B, you can see there is no room. There is no room where like light can enter. Like, but in the building A, you can see it's made up of glass. So, in the building A, you can see it's made up of whole glass building, so light can enter. In the building B, light, a very little light can enter. So, if you have to put your small saplings in a room, I would like to choose A, right? Because lot of light can enter in this building. So the picture B that is in your screen is from is very old picture, as you can also see. So it's a very old picture of around 1800s, and like around 1800 picture is it. This is an old botanical garden in London. So you can see people, they don't have understanding of the relationship between the light and plants. They have no knowledge about that. So there is very less understanding of light. So, but there was one guy named Van Helmont. So this guy, he likes plants a lot. So he was understanding about the plant during the 1600s. So he really liked plants. So before him, around 2000 years ago, people, they thought that trees, they grow by eating soil. So what he did, so he designed an experiment, and in this experiment, what he did, he took soil, he took a big pot and a small plant. So what he did, he weighed a small sapling of plant, as you can see in the screen in the overhead camera. What he did, he took a small plant and he weighed this small plant, and he weighed the soil, and he put the soil and the small plant in a very big pot. In the next for the next 5 years, what he did, he just gave water to that small plant, and after 5 years, he repeated the same experiment. What he did, he weighed the whole plant and the soil. So surprisingly, what he see, that the soil, which was 5 years ago around 90 kilogram, the soil was still the 90 kilogram, but the tree has grown from 2.5 kilogram to 76 kilogram, but the weight of the soil was the same, around 90 kilogram. So through this experiment, he understood that plants don't grow by eating the soil. So this was his experiment. But it's not very people were not aware of how plant grows. So as we can nowadays, we can very easily say that the plants, they do photosynthesis, but it was not very obvious in the past years. So today, as I've mentioned, we will learn a few things about the photosynthesis. So you can take your things that we have mentioned, as you can see in the PPT. You can take all these things in front of you, the things which I have mentioned. You can see in the screen, or you can just take out everything. we can start the experiment, right? Everyone has 3 syringes. You have 3 syringes. You can just raise your hand if you have 3 syringes. KGVB Langlol. Yeah, I can see that. Great, amazing. So we can start the experiment. You have the baking soda with you, right? So what we'll do, I don't know whether you have a weighing balance or not, but if you have a weighing balance, then it's very good. Otherwise, you just need, if you have a spoon, if you have a spoon with you, if you have weighing balance, that's very good. If you don't have, then I can tell you how much amount of this baking soda you will need. So you will need around 4 gram or half of this teaspoon. If you have this this spoon, you can put half of your baking soda. You can measure it, or you can just, so you can see if you can take this amount of baking soda, it will turn around 4 gram. See, oh, it's just a little bit of more. I will add. You can yes. So you just have to measure half of this half teaspoon of baking soda. This much amount of baking soda you need if you don't have the weighing balance. Okay. I can just put it here. Is it okay? If you have any question, you can write. The first thing you just have to weigh 4 gram of baking soda and put it in one of the cup. Everyone is with me? Have you done the first process? Just have to weigh the 4 gram baking soda and put it in one of the glass or whatever you have. It's done. KGBV Langlol, Sunita, KGBVG. Okay. Rohika Shani, do you want to ask anything? RUQUIA. You want to ask something? Oh, it's done? Amazing. Because there are a lot of schools. Okay, good, good. So I'll move on. I'll move. Okay, good. Okay. So what the next step is? You have to add 100 ml of water in this powder. 100 ml. Okay, I will. Okay. You just have to add 100 ml of water in this powder, baking soda. And you can mix it. Okay. 100 ml is like, is this much. Like, it's not very, if you don't have a measuring cylinder, you can, I will show you the bottle that I have used and how much, like, it is just, like, from the bottom, it is, I think, this much. It's 100 ml from the base. Okay. Yeah. I am happy that you are raising your, I can mix it with the spoon. Okay. Amazing. And so mine one is already mixed. So in the second jar, I will add 100 ml plain water. Okay, I don't have to add any of the powder. I just have to add 100 ml of the water. Okay. And I will mark them as, I can write it baking soda. I am adding just the initials of baking soda or B, and this one as a water, W. Now you can write anything A or B, but you just have to remember in which you have put the baking soda and which one is just a plain water. Okay, good. I can see kids are doing KMM Singaria. They are doing it. I'm very happy. Good. Okay. I can see which other school is doing it. Okay. Wow. KGBB Langlol. Yeah, I can see you. So what I'll do, I'll keep you in the screen along with me so that I can it will give me a sense that okay, so people are doing work with me. Okay. So I have written the initials. Oh, I'll go overhead. Okay. So these are my two jars. Okay. Right. So you have 3 syringes with you, right? What you will do, okay, what you will do, you will just have to remove the plungers. You have to remove the plunger out of the 3 syringes. We remove the plungers, okay? And you will mark them 1, 2, and 3. Mark them 1, 2, and 3. Just mark 1, 2, and 3. Okay. Sunita ji, KGBB, I can see that you are doing. You can mark 1, 2, and 3 in your syringes. Okay. Is it done? Okay, good. So now, as I have told you to collect the leaves also. So now, as you also have, you have also seen that I have, just hold on a second, right? Okay. So as I mentioned that you have to collect the leaves and you have a straw also, right? This straw. So what we will do now, we will make small discs out of these leaves. So what you will do, I can also uh, show in the PPT. You have to make these holes out of leaves. Okay. How you will do it? You have to put your this straw on the leaf and you have to press and rotate it. When you will rotate, you can see there will be a hole or you can just take it out as a disc. Okay. What you have to do, you have to make at least 15 disc out of your leaves. Okay. Is it okay? You have to make at least 15 discs out of your leaves. Okay. You can distribute the leaf, or if you have more than one straw, you can give it to other students. Okay. I will also make discs with you. I will also do the experiment again. And I really like making these discs whenever I do this experiment. Amazing. So if, you know, at times what happen, your discs will go inside the this pipe. So what you can just what you will do, if something happen, you will just blow and the disc come out. Okay, you can do this. Go ahead. Okay. Let's see who makes more discs, you or I. I have 4. I have 5. So it will be good if you can make the round discs. Okay, I have made 5. If you have made your discs, any school which he has completed the making disc, you can raise your hand in the screen. Okay, wow. Just give me your first. When I was of your age, I also do lot of things very fast, but now I am old. It will take some time. 21 made. Wow. They have made 21. Good. So while I am making the discs, what you will do, you will put like 5 to 6 disc in every syringe. As I can show you, you have to put at least 5 or 6 disc in every syringes. Okay. As I have put it in the 1 or 2, I will show you over the PPT also that you have to put your disc in the syringe. Make sure all the discs, they go at the bottom. Okay. Make sure all of them go to the bottom. Okay. While you are making all other discs, like you are putting the disc, I will take out my discs, remaining ones. Okay. Good. Wow. Give me 1 more minute. Okay. I will add 1, 2, 3, 4. I just have to make 3 more discs, then I can start the rest of the session. 1 more. 1, 2. Just give me 1 second. 1 more minute. By that time, you can see my face, how difficult it is for me to take out these discs out of this leaf. Okay, so I know I am last, but I have 1, 2, 3, and 4 discs. Okay, now you can see I have discs in my syringes. Now what you have to do, you have to put your this plunger in every syringe again. Make sure you don't have to press any of the discs. You just have to, and you have to like keep this space in your disc, this much space. Okay, in 1, 2, or the that you have numbered 1, 2, and 3, you have to put the plunger back and make sure not to press the discs. Okay. Okay. Have you done the? Okay, amazing. Now you have to listen very carefully. I'm giving you a secret. Okay. So you have 3 syringes, 1, 2, and 3. Okay. So, okay. So in syringe 1 and 2, in syringe 1 and 2, you as we have already make the solution, right? These are my 2 solutions. So in syringe 1 and 2, you have to add the solution in which you have mixed the baking soda. I will again repeat it. In the syringe 1 and 2, you have to add now, we will do together, but listen to my instruction first. In the syringe 1 and 2, we will add half of this solution, which has baking soda, and in the syringe 3, we will add we will add just the water. The beaker which has only the water, we will add in the syringe 3. You understand? I will again repeat. In the syringe 1 and 2, what we will add? We will add the solution which has baking soda, and in the syringe 3, we will add only the water. Okay? Is it easy? Amazing. Great. Now, fill half. Don't fill full, just fill the half. Okay? As you can see, I have filled 1 and 2 with the solution that has baking soda, and the syringe 3, I have just added the solution which has water. Okay? Have you done? Okay, I will again repeat. In that you have 3 syringes. In syringe 1 and 2 that you have already marked 1 and 2, you will add the solution. You can fill your syringe 50%. Don't fill it fully. You will add solution that has baking soda in syringe 1 and 2. And in this third syringe that you have marked 3, you have to add the water only. We have 2 beakers with us. 1 and 2, we will add the baking soda, and in the syringe 3, we will just add the normal water. Okay, I will again mark them because that was not a permanent marker. So 2, okay, 2 and 1. Everyone done? It's done? Great. Now you can see, if you just hold your syringe like this, you will see all of this, all of your discs are at the top, as you can see in the PPT also. I'm just sharing. All of your discs are floating at the top. Okay. So why are they floating at the top? Anyone can answer in the chat box. Like, what do you think? Why these discs are floating at the top? As you can see in the PPT also, all of your discs are floating at the top. Anyone can write? Is there anyone who can tell me why these discs are floating at the top? Anyone knows the answer? I will tell you. So we all know that plants, they take air inside, air has carbon dioxide, a lot of things. So to do the photosynthesis, plants, they take oxygen, like they take air inside in the leaf through stomatas. So there is a space within the leaf. Within the leaf, plants have small space in which between the cells in the leaf, they store the air, so that they will use this air during the process of photosynthesis. But there are small gaps within the cells in the leaf where they will store the air, because in this leaf we have air. Because of that, these leaves are floating. Okay, all these leaf discs are floating. Some of you are still doing it. You can do it, no problem. We will move together, no problem. Okay, I'll again give you the instructions. In the disc, in the syringe 1 and 2, you will add the solution that has baking soda in it, and in the syringe 3 you will just add the water. Okay, it's done. It's done. It's done. Okay. Not hearing. Great. So now we will have to remove the air that is present in the leaves. It's a very big task, to remove the air from the leaf, but I will give you a very simple and easy way to remove. Has anyone gone to a doctor? I am just acting like a doctor now. What you have to do, you have to, as you can see, in the here, you can see there is a lot of gap. There is a water, there is a gap. What you have to do, you have to push the plunger and you have to remove all the air that is at the top and make sure one drop came out. Okay. All the discs are like this. See, now there is no space here, right? What you will do now, you have to put your finger here and you have to press the plunger downwards. Okay, like this. You have to push the plunger downward like this. I will tell you what will happen while doing this. You just have to put your finger here and push the plunger downward. Make sure all the leaves are still. If your leaves are at the top, you can just shake this this vial and make sure all the leaves, they are in the water, and push the plunger backwards. Slowly what you will see, and you can also tap. You can slowly see, as you can see here, your leaf discs are going downwards, as you can see here. I'll do it again. I just have to push it so that all the air that is trapped at the top, it will come out. Just one drop coming out like this. One drop is coming like this. Now put your hand here, push your syringe backwards, this plunger backward like this, and slowly you can see, just tap it. As you can see, all the leaf discs, they are now at the bottom. See, they're at the bottom, except this one. This, oh, this is also gone. Okay, so do it for all the 3 syringes. Is it doable? Anyone facing any problem? I'll repeat it, okay, with you. I have my second syringe with me. What I will show everything in the overhead, no problem. Okay, so see, this is my second syringe. Okay, leaves this are still at the top. I will push it so that the air that is trapped out at the top, it comes out with the like. Now I'll drop. I will put my one finger here and I will push my plunger backwards. Make sure all the leaves are still in the water. Okay. And you will remove just tap it. You can see the leaves are sinking down. I will repeat it again. Make sure every single disc comes at the bottom. Okay. Make sure some one of you might be interested to become a doctor. It is just a small training. How to put injection, right? If you have to make sure that every single disc has to come at the bottom. So I'll do it again. It will take some time, not very much. Make sure every single disc in the injection vial comes at the bottom. Wow. So my second syringe, all the discs are at the bottom. I will show you here. See, they are at the bottom. The first one also, second one also, all the discs are at the bottom. They are not floating. I will do for the third one also. Okay. I'll just squeeze it so that the air at the top, it will. Now I will put my finger here and I will push it backward. Make sure all the leaves are still in the water. I am just holding it horizontally because I have to show it in the camera. You can do it vertically like I am doing here, right? Wow. My leaves are very obedient, I think. They are all. Wow. Yes. I have all the leaves, discs. They are now at the bottom. Wow. I am first. My all discs are at the bottom. Yes, I am first. You can do it. Take your time, no problem. Okay, wow. Okay, KGBB Moriang, Manipur. You don't have stuff with you? You don't have material? You can unmute KGBB Moriang. Hi. You don't have material with you? You don't have material? Okay. You can see. No problem. Okay. Wow. Rajintha KGBV Raikode. Ah. So you are done? Raikode KGBV. All the discs are at the bottom. Wow. Congratulations. Thank you. Good, good, good. Nice. Okay. So if you are done with this step, we will move forward. So now, as you can see, what happened, all the discs, they are at the bottom, right? One of my discs came up. I don't know why. I'll make sure that, no, it's still, yes, it's at the bottom. Okay. It's very important to do this step. All the discs have to come at the bottom. Okay. They are not at the bottom. KGBB Langlol, you can take your time. No problem. No problem. Take your time. You can do it. If you want me to repeat the process, like I can tell you, you just have to, like, first of all, if there is any space over the water, remove that space, remove that air by slowly pushing the plunger. You know, if there is a space like this, there is an air, just have to push the plunger very slowly, very slowly, very slowly. There is one drop, it can come out. Put your finger here and push the plunger downwards slowly. What happened in this process? Why discs they came at the bottom? The reason is when you apply the when you push the plunger, the pressure inside decreases and all the air, it comes out from the leaf. And when all the air that was earlier in the leaf, it comes out, it will replaced with the water. It will make leaves heavier. Because of that, all the leaves, they comes to the bottom. Okay. Now all the leaves are at the bottom. All the discs are at the bottom. What we will do, the next step is, so these are the 3 syringes that I have. All the syringes are at the bottom. Now, as you see, my syringes are half filled. 1 and 2 and 3, they are half filled. Now, fill these syringes fully. Syringe 1 and 2, we will put the same solution, the solution which has baking soda. We will add it fully. Syringe 1, I have put it's fully. No, it's full now. All the discs are at the bottom. Same we will do it with the syringe, second syringe. You can see all the discs, they are, few of my discs came up. I'll repeat the process with these, but you have to fill all the, if your syringes are at the top, you can just tap. I think there are few air bubbles attached to the leaf. That's why they are at the top. Now see, you can see slowly they are coming downwards. And in the syringe 3, you will fill it with the water only. Syringe 1 and 2, we will fill it fully with the solution with the baking soda. Syringe 3, we will fill it with the water only. Okay, if your discs are at the top again, you just have to repeat the process and make sure all the discs, they comes at the bottom. Okay, you have to make sure every disc has to come at the bottom. My few of the discs, they are again at the top. Wow, now they are at the bottom. Okay. I'll again repeat, in the syringe 1 and 2, we will fill it fully with the solution with the baking soda. And in the third syringe, we will fill the syringe with the water only. Okay. I don't know why my discs are, okay, now everyone at the bottom. So you can see, and because it's a green. Okay. Is it done? No? Okay. School, Mizoram school with the yellow shirt. Is it not done? No? We will do it together again, okay? Your discs are at the top, still at the top? Yes or no? They are at the bottom? They are at the top? Your discs are at the top, still at the top?
Yes.
Okay. So do it with me, like push your syringe slowly so that if there is any space, like is there any space at the top, just have to push slowly, slowly, slowly so that all the air that is at the top, it goes out. Put your finger at the top like this. Now just push the plunger downwards. Make it 4 or 5 times. Slowly you can tap your, see? Slowly all the leaves, they will sink down. See? You can see my discs are, they are coming down. Slowly, slowly, slowly. If you can, if one of you can come close to the camera and show me how you are doing, I can suggest you.
Yes, sir.
Wow. You can come to the camera, no problem. Don't shy. I'll not eat kids. I am vegetarian, purely vegetarian. Wow. Now push it slowly. Push it down like from the bottom. Yes, yes, yes. Now put your one finger at the top like this. Now, yeah, push it. Push the plunger downwards. Yes, push it now. Okay. What you will do now? Tap it. See? Are they coming downward? No. You will repeat the process. You like make sure all your discs, they come downward. Make sure. Okay. While you are, pushing your syringe downward, make sure all the leaves are still in the water. Okay. Then make sure all the leaves, while you are pushing your plunger downwards like this, make sure all the leaves are still. Make sure all your leaves are at the inside the water. Push it like this. No? Remove the air that is at the top. Hold it like this. And just tap so that, oh wow. Okay. So whosoever is done with the this process, wow. So my all the leaves, they are at the bottom. As you can see here, this one is just going downward. All of them are at the bottom. Okay. Wow. I will put it my okay. Which school has done? Which school has all the discs at the bottom? Is there any? Oh wow. KG. Okay. Moriang. Okay, I will. Which school has all the discs that are at the bottom? Is there anyone? You can just raise thumbs up. Oh wow. Okay, so I will put you in the spotlight. Great. KGBB Raikode. Good. Now, you have the aluminium foil with you, right? You have the aluminium foil? Show me the aluminium foil. This is aluminium foil or what? This is a transparent thing. Aluminium foil. Yeah, that is aluminium foil. Oh, I don't know. Like my aluminium foil is like this. I don't know what. It's a transparent thing. It's not aluminium foil. If you have the aluminium foil, which school has aluminium foil? Okay, I will. I can see one of these school has. Which school has aluminium foil? Wow, good. I will spotlight. Good. KGBB Urban Wow, good. What you will do, you have to like you take your syringe that has number 2 on it. Take your syringe that has number 2 on it and cover it with the aluminum foil like this. This is my number 2nd syringe that has baking soda with me. What you have to do, it's very cold out for this syringe. We have to cover it with the aluminum foil like this. Okay, can see it? Yes. Make sure no light enters, like there is no space from where light can enter. Make sure. And you have to do it with the syringe that have marking number 2 on it. Okay. Make sure not to push it and make sure while you are doing this, all the discs are at the bottom. So this is my 2nd syringe. All the discs are at the bottom. I am just putting it top of it. I am covering my number 2nd syringe with the help of this syringe. Okay. Good. Few of the students are doing it very good. Okay. I will add another school, Swarupa Kate Palli. You have to cover it fully. The slide, the syringe which has 2nd number on it, cover it with the, yes, just cover it. Don't, yes. only the syringe that has number 2. Swarupa Ketipilli school, like I can see 2 of the girls have these syringes that are covered in the aluminum foil. Make sure only the syringe which has number 2 on it has to cover in the, let's see. I have 3 syringes with me. I have 3 syringes with me. Okay. I have 3 syringes with me. Okay. My numbers, they are 2, 3. You can see, I'll just show you. I have 3 syringes. Number 1 and number 3, they are still not covered. Only the syringe that has marking number 2, that is, I have covered it in the aluminum foil. Okay, it's done. You can just say, you can just thumbs up if it's done. KGBV, you can see beautiful faces. Okay. Yes, KGBV, Chintapalli. Is it done like this? No. Syringe 1 and 2, like syringe 1 and 3, they are not covered in the aluminum foil. Only the syringe that has number 2, you have to cover it in the aluminum foil like this. I will show you it in the overhead like this. This is my, I'll remove it. 1 and 2, 1 and 3, sorry, 1 and 3, they are not covered in the aluminum foil. Only the syringe that has number 2, I have covered it in the aluminum foil. Okay. I can also show you on the PPT. Now you can see syringe 1 and 3, they are still open. Only the syringe with number 2, it is covered in the aluminum foil. Is it done? Okay. Is it done? You can just raise your thumb if it's done. It's done or not? Wow. KGBB, good. Langlol, Manipur, good. I like it. So now if you have a beaker like this, you will put all of your like syringes in it. I will show you. Okay. If you have a stand, we had made some stand. So like put this like this. Okay. I'll keep all the things away. You just have to put all your syringes in a jar. You can throw the water and you can put your syringes in a beaker. Okay. Now, what you have to do now, what will you do? You will switch on the light. I will just switch on the light. So you can see, I have just switched on the light. You can take your time, no problem. I'm again switching off the light. If you have a light source with you, you have light source. I have no idea like whether it will be done with the mobile light or what, but if you have a light source, do you have light source with you? Okay. Any of you have a light source? Amazing. So I'll just put this school, KGVB Common Urban. I can see that there is a light, glowing light. Good. Can you just switch off your light for a minute? KGVB Common Urban. Oh, great. Can you show me your these 3 syringes? Can you take out and show me? Yes, all the discs are at the bottom, right? Okay, good. Now put them again in the bottle, in the glass. Thank you. Thank you very much. Okay. I think most of these schools have already. Okay. Now, as you can see, all the syringes are in a jar, in a glass jar. Now I will switch on the light. Good. I have switched on the light. You will also switch on your lights and keep your this jar very close to the light. Okay. Keep your lights very close to the light. Make sure the aluminum foil is and try to observe what's happening. It will take like I think 2 or 3 minutes. Wow. You can see. Just have to observe slowly what happens to your syringes. If anyone see any change with the leaf discs, you can write in the chat box. Just observe them. Observe them. What's happening to your discs? Like it will take some time. It will take 1 or 2 minutes. We will observe. I will also observe my discs. Okay. Meanwhile, I am just unmuting one of the school from Manipur. They are doing very good. KGBV Langol. Langol. You can unmute yourself if you can. KGBV Langol. You can unmute. I can. Unmute yourself. Okay. I don't know if internet is not working fine or what. I will pinpoint some other school that are doing good. Okay. So this is Ranjitha KGBV Raikode. Okay, good. You have torchlight, no problem. Okay. You just have to put your syringes in a beaker and put the light close to the beaker. Put the light very close to the beaker. Wow. There is one more school out here. So I will explain what's happening here. Okay, wow, good. I have spotlighted one of the Mizoram school, Samsung SMT735, girls with the yellow shirt. Yes, so they are doing very good. So they have put all the syringes in the beaker and they have put the light. Is there anyone who has seen any changes with their discs? Anyone? Anyone? Something happened to your discs? Nothing? You can just write in the chat box. Okay, so maybe it will take some time. I will show you what will happen. Oh, wow. There is one school, Kondur Umma Devi. Wow. There is one school who has, yes. Oh, wow. So this school, they have said that something had happened to their discs. I am unmuting you, Kondur Uma Devi ji. You can unmute and tell us what happened to your discs. What happened to your discs? RN Kondur Uma Devi ji. If you can tell what happened with your discs, you can unmute and tell us. Okay. Okay. So no problem. What I'll do, I'll share a small, wow. I will just, something had happened. You can see in my number first this syringe, you can see one of the discs. I'll just switch off my light for a while and I will show you. You can see one of the discs, it come at the top in my syringe, which has number 1 on it. You can see one of the discs is at the top. I'll just switch on my light again, and you can see. One more syringe is, one more disc is coming upward. See, it is coming upward, right? Here. It is coming upward. Yeah, I'll just show you. I'll just switch on the light again, and we can see like if something happens. Two of my discs that are in the syringe one, they are at the top, at the like they came. Yes, I'll just hold on for 1 minute, and I will show you. Wow. Is there any other school whose discs they move towards upward? They move upward. Is there any school? Any other school whose discs move upward at the top? You can just observe bubble in the third one. Wow. You have good. That's a very good observation. KGBV, Khaman, Urban. That's a very good observation that you can see few of the bubbles. You will see the same bubbles with your discs that are in the syringe one. Oh, you can see now. See now. I will just switch off the light. You can see these ones are moving upward. See, slowly. You can see in the screen. One of the two discs, they are coming upward. Wow, at the top. See, and you can see also small bubble. There is a small bubble attached to this leaf disc. See, it's come upward. Wow, amazing. Good. Wow. I think my okay, good. That's nice. So what I'll do, I will show you a small video that I have made. In this video, you can see I have 3 jars. You can see 2 of the jars very easily, but the third jar, it is under this cardboard box. I'll just play the video. I'll just see at the right beaker. You can see all the discs. They are slowly moving upwards. See, in one of the beaker. Oh, see how they are coming upwards, right? See, it's a magic. See, all the discs in one of the jar, they are moving upwards, but in the second one, that you can see all the discs, they are still at the bottom. Now I will just fast forward it, and I will show you. You can see I will just remove the cardboard one also. I'll just see. I have removed the cardboard one, cardboard also, and you can see all the discs, they are at the bottom. I will show you one more video. You can see the movement of discs. See, slowly the disc at the bottom. See, if I just pause this video, you can see there are small bubbles attached to this leaf. There are small bubbles attached to this leaf in the video. While the discs, they are moving upward, you can see there are small bubbles that are attached with the leaf. Wow. Wow. So is there any school whose leaves are like they are at the top, like the one that I have? This is my number 1 syringe. Some of my discs are at the top. You can see. I'll just switch off the light for a while. I'll show you. See, some of my discs are at the top. Okay. Some of my discs are at the top. Is there any school whose discs, okay, they come upwards, they are at the top? Is there any school? You can just write in the chat box if same thing happened to you also with you. I'll just switch on the light again to keep going the experiment. Is there any other school whose discs, they come upwards, they move upwards? Okay. So now what I'll do, I'll show you all the discs to see. These are my 3 syringes. I will hold like this. So this is my number. I'll put them in the order. Okay. So this one was my second. This is my third. So see, this one is still moving upward. The number 1. It is going upward. But as you can clearly see, I'll just put it like this. So in the syringe number 3, my discs are still at the bottom. Now I'll remove this aluminum foil and we will see what happened inside this syringe. Are you ready? You are excited to see what's happening in the syringe that is covered in the aluminum foil? Should I remove it? Should I remove or not? Should I remove? Okay, so what I'll do, I'll remove the syringe that has aluminum foil in it. I'll remove it and we'll see what's happened in this syringe. What do you think? What happened? What do you think? Is there anyone who can write in the chat box who can explain me what I can see in the second syringe? All this, do you think the uh, leaf discs, they are at the bottom or they will move upward? What do you think? What will happen in the series number 2? Anyone can write in the chat box. Any guess? Before I'll remove the aluminum foil, can you write in the chat box what you can observe? What will happen in the series number 2? The discs, they are still at the bottom or they have moved upward? What do you think? Write in the chat box. Meanwhile, I'll remove the number second. No change. Wow. So see, this is my disc number 2, and you can see all the leaf discs, they are still at the bottom, like the one in the syringe number 3. See, only the leaf discs that are in the syringe number 1, they move upward. Rest of them, they are still at the bottom. Is there anyone who got the same results as I got? Is there any other school? You can write in the chat box if you have the same results. You can just write in the chat box. Okay, I think no one has got the same results. So what we will do, so before this, I will explain. Do you know what happened? Is there anyone who can explain me, or you can write in the chat box or in the Google, or sorry, in the YouTube, that what happened in all of these syringes? Why only the leaf discs that are present in the syringe number 1, only they came upward, rest of them, they are still at the bottom? Is there anyone who can explain what happened? You can write, or you can just raise your thumb so that I unmute you and you can explain me what you have understood so far from this experiment, like what happened, why only the leaf discs in the syringe number 1, they are all at the top, but rest of them, they are still at the bottom. The leaves are same in all of these syringes. There are only few differences. Based on that difference, can you explain me what happened in the syringes? Anyone who can raise your hand, like you can raise your hand in the Google, in the Zoom. I will ask you to unmute and you can explain me. Okay, no problem. So I'll explain you what happened in this experiment. So in the syringe number 1 and syringe number 2, we have provided everything. Like all this, the solution is same in the syringe 1 and 2. It has solution which contain this sodium bicarbonate or baking soda. If you go and check, whenever you have time, you check what is the chemical formula of this baking soda. You will get to know that this baking soda has lot of carbon in it. And when we dissolve the baking soda in the water, the carbon, it dissolve in the water. So the discs number 1 and 2, they have source of carbon in the water. And in the syringe number 3, we have not added this baking soda. So it doesn't have any carbon source. Okay. And in the syringe number 1 and 2, in between these two syringes, I have put this aluminum foil in the syringe number 2. Okay. And the rest of the syringes, they were exposed to the light. So only the syringe number 2, it doesn't have any source of light. The light is not entering through this aluminum foil. So what's happening here? Imagine these discs as trees. One of the tree, the syringe number 1, we have provided them with carbon, water, sunlight, and the leaves, they have pigments in it. In the syringe number 2, imagine this tree, everything is there, but we have kept this tree in the shade. No light is falling on the leaves of this tree. And in the syringe number 3, it has everything except there is no carbon given to this leaf. Okay. So what happened here? In the presence of light, in the presence of water, in the presence of pigments, and in the presence of carbon dioxide, what happens in syringe number 1? Photosynthesis happens. And during the photosynthesis, as we all know, that plant, they release gases. And we know that in this process of photosynthesis, plant release oxygen. So while these leaf discs, they were moving upward, they have small bubbles attached to the leaf discs. If you observe carefully, you can see small bubbles, they are attached to the leaf discs. So those bubbles are of oxygen. And when they are attached with the leaf, they will make these leaf discs lighter. When bubbles are attached, they are more lighter and they move with they move upward. So only in the syringe number 1, photosynthesis is happening. In the syringe number 2, we have not provided the carbon source. And in the syringe number 3, we have not provided, sorry, in the syringe number 2, we have not provided any light source, but there is a source of carbon dioxide in it, but light is not entering in it. In the syringe number 3, everything is there, but there is no carbon dioxide in this syringe number 3. So you can clearly see the role of carbon dioxide. What's the role of carbon dioxide in photosynthesis? What is the role of light in the photosynthesis? If there is no light, no photosynthesis happens. If there is no carbon dioxide, no photosynthesis happens. If we provide carbon dioxide, water, light, in the presence of all these things, pigment present in the leaf, they will do the photosynthesis, as we can see in this experiment. Okay. Is there anyone who wants to ask anything? Is it clear to you what we have did in this experiment? What we will do, we will try to do this experiment again in our next session. Okay. If you have not got the results so far, what we will do, we will repeat this experiment again in the next session. Okay. Is it fine? You like this experiment? Is it very easy or difficult for you? You can write in the chat box if you feel this is easy or difficult, or you doable, or you will not able to do it, or you want to repeat it again. Oh wow. Suman, they said it's very clear. Good. I'm very happy. Is there anyone who we'll do it again in the next session? No problem. Okay. We will add some variant also. As you can see in this experiment, what we have did, we put only 1 tablespoon. I know why my camera. Okay. You can see we have put the sodium bicarbonate. We will, in the next experiment, we will reduce the amount of bicarbonate. We will use different light. I will let you know what all things that you need in your next, in the second session. But wow, some of you have said it's very clear. Wow, someone is asking, we have not provided the carbon dioxide, but we have provided sodium bicarbonate. It's very good question. It's very interesting question that you have asked. Amazing. If you want to ask any question, you can write in the chat box. We will discuss all these questions next time. It's very, this experiment is very amazing. You know, when I did it for the first time, I did it in like 3 months ago. And when I did this experiment, I was very excited. I thought if someone has taught me this experiment when I was at your age, I would have understood this photosynthesis very easily, because it's a very easy, very amazing process. But still, if you have not understood anything, you can write in the chat box. It will help me a lot to answer your question next time. So I'm just I will take a picture of the question that you have asked. It's very amazing that you are asking. Very good question. If anyone of you have any question, please write down in the chat box. Okay. So before I'll just move, I will take 5 or 6 more minutes. I would like to ask one question from you. Question 3. Okay. So suppose if all the trees and plants, if they disappear, suppose something happens to them and all the trees and plants, they disappear from the earth, what do you think, which creatures will find it difficult to survive? What do you think? I'll repeat the question. Like, suppose something happened to the earth and all the trees and plants, they get disappear from the earth. What do you think, which creatures will find it very difficult to survive? Do you think it's very difficult for us to survive without the plants, or lizards, they face the problem, or bees or goat? What do you think? Who will face the problem? Or all of them will face the problem if all the trees and plants, they disappear from the earth? What do you think? Bees, you know, bees, they do a lot of pollination. Goats, they eat tree. I don't know what's with the lizard. If something happens to plant, I don't think something happens to the lizard. But still, if you can answer, what do you think? Who will face, which creatures will find it difficult to survive? I'll give 10 more seconds. Okay. Wow. So I am ending the poll in 10 more seconds. Okay, I'm ending it. Okay, good. Amazing. So we will repeat this experiment next time, and I will make some changes in the next experiment. So I'm very happy that at least you were able to do few of the things during in this experiment. You know, I do this experiment with lot of teachers, like you are kids. I also do this experiment with lot of teachers. Even they were not able to do this part. It's very difficult for them to do. Okay. So don't feel ashamed that, oh, you were not able to do it. Even a lot of teachers, they were not able to do it. Okay. So we will do all this thing in the next session. But what you will do in this, like now I want to give you some homework. So what you will do in today's homework, that I'll just share, I will share this one PPT. Okay. Okay. What you have to do, as I have shown this slide earlier to you, there was a person on named Helmont, Van Helmont. What he did, like in his experiment, what he did, he tried to understand the role of soil, like how do plant grows. So what he did in his experiment, he took a, I'll just take this one. He took a small pot and a small sapling that I will show you in the overhead. This is a small sapling, and he took some mud also, like soil, right? He took some soil and a pot, a plant, and soil. Okay. What you have to do, you have to repeat this experiment, what Van Helmont did. So what you have to do, you have to take a small plant, not very big plant. You don't have to go to your teacher's house and take his or her, this pot. You have to take out a small sapling, and you can take a broken glass that don't have a handle, or if you have a pot with you, that's very fine. One pot, you have to take one pot, you have to take one small plant, and you have to take some soil. And before starting the experiment, what you have to do, you have to measure, you have to weigh. You have to weigh in a weighing balance. Your school must have some weighing balance with you, like this kind of weighing balance or a bigger one. What you have to do, you have to measure the weight of your small plant. You have to measure the weight of your plant. You have to measure the soil. Okay. And what you will do, you will put the soil in the pot, and you will put your plant in it. Okay. And for the rest of your, like, I think you will take around 10 or 15 days. What you will do, you will just give that plant water. Not very much, like not very, like you don't have to uh, put all your bottle in the pot. Just little water you have to give for at least 10 to 15 days. After 15 days, what you have to do, you have to repeat the same process. What you will do, you will take out your plant very easily. You will take out your plant, you will measure it weight, you will take out all the soil, and you have to measure it, and you will compare the data, like what you did in 15 days ago, like maybe today. What you will do, you will make a notes, like, okay, your plant, your small plant, it weighs, or it weigh around 15 gram, let's suppose 15 gram, as you have put 100 gram of your soil. Okay. And what you will do, you will put everything in the pot. Okay. And rest for like for the next 15 or 20 days, what you have to do, you just have to put little water, not very, like not you have to do, you don't have to put a lot of water, just a few ml water is enough for this plant, small plant. And after 15 or 20 days, it depends on you, 15 or 20 days, you have to repeat the same experiment. What you have to do, you have to measure the weight of the plant, and you have to measure the weight of the soil, and you have to share everything to us. Okay. Is it doable? It's a very simple, easy experiment. Okay. I will see you next time with some of the variants that we have did today, and we will again go through this process and try to understand this concept of photosynthesis in amazing way. Okay. Should I say goodbye to all of you, or is there anyone who wants to talk, who wants to discuss? Just write in the chat box if you want. If you don't want, can leave the chat box, and I will see you next time. Okay. Bye bye. Bye bye. Oh, wait a second. Wait a second. One of my insect box also wants to say bye to all of you. So these my insects, small insects, they are saying bye to you. Okay. We will see you. I will see you next time. Bye bye. Take care. All the best.